Hi, my name is Matt Anderson. I'm an associate professor here at Ohio State in the Department of Microbiology, as well as the Department of Microbial Infection Immunity in the medical school. Before I begin, I just want to acknowledge the fact that Ohio State currently sits on Eastern Shawnee, Wyandotte, Miami, and Potawatomi land slightly to the north, and that this land was acquired through the Treaty of Greenville, fundamentally under duress from a number of tribal nations under threats of violence from the US government, and that Ohio State continues to profit financially from the sale and the interest collected on these lands. So what I'm hoping to walk you all through today is how really we need to move from performing research in biodiversity that includes indigenous people or indigenous contexts of biological systems from academia and governmental research into tribal nations and other indigenous communities. And I start by really talking about the false primacy of humans and how the view taken within ecological contexts can absolutely change your understanding of what's going on. So we center a lot of protections that exist within human-based research. This exists for institutional review boards as well as human subjects protections. What we don't do is we don't often put those same regulatory controls in place for other kinds of entities at the center of research, either animals, different aspects of the environment, including those that are both biological and non-biological. And this really stands at odds with our understanding of the relationship that exists between humans and other aspects of biological and non-biological life. So if we look at a standard ecosystem type framework, we can see within this picture of the forest, we have both different plants, including ferns, trees, and a number of low-lying shrubs that are all interacting with non-biological entities with their environment. The environment is shaping the growth of those plants, and the plants are absolutely shaping the contents and the composition of those non-biological components of the environment. If we scale down a little bit, we see ecosystems embedded within ecosystems, right? Within a microbial community that could be found, for example, on a tree, we see additional relationships that exist between different microbial entities at this scale within the larger ecosystem that we can see by eye. So if we think about this within the context of my microbiome, where a lot of my work has been centered, this really involves a relationship that exists between those things that we can see oftentimes under the microscope, fungi, protozoan, and bacteria, as well as viruses, something we often neglect within these kind of ecological niches. Now, the way we look at these communities and the way we look at this data is really going to determine our understanding of it. If we look at it based on the number of organisms, we say clearly viruses are the dominant players within this ecosystem framework. If we look at it based on DNA, though, we would say bacteria are clearly the center of all these interactions. If we look at it based on the mass of these organisms, we may give a more convincing role toward fungi and protozoans because of how large they are compared to either bacteria or viruses bacteriophage within these systems. So an intimate understanding of the ecological niche is required here. Otherwise, a simple glance at it one way or another could lead you in the wrong direction, potentially depending on your question. So we can see this also not only with kind of static contents, but also within relationships. Here I'm showing a very quick relationship between three different microbes that are found within the human gut: Lactobacillus, Enterococcus, and Candida albicans, a yeast. And either are mutualistic relationships defined by the green arrows or antagonistic relationships defined by the red arrows. But if we begin incorporating other aspects of the ecological system here, we see that these relationships get a lot more tricky to decipher. It's not quite that simple. So it requires a really intimate knowledge of the system to be able to appropriately decipher what is going on. And so if we're thinking about that within either microbial ecology research or other ecological contexts, we need to really start by removing the centering on the individual human. We need to be thinking about consenting an individual, consenting a community, and consenting a set of communities within this work. And this, begins to acknowledge relationality that exists between different people groups. Working with one specific population does not mean that that work will not impact another population. So we need to be thinking about how the broader context of the work will be impacting more than one community. We also need to start thinking about the way that we have responsibility to prioritize the interests of communities over academic research interests and be responsible to those communities to carry out the work under exactly those guidelines that have been given. This is important, especially to regulate this data, because data from one human or human-associated population, if we're dealing with non-humans, can inform a human population and potentially other human populations. So we need to be thinking about how defining context for people tells about people. And really, I think this requires a movement to promoting Indigenous capacity. I think that as academic and government researchers, these environments are not ideal for being able to produce knowledge around Indigenous peoples adequately. And I think we've demonstrated this because we as a scientific community have been unable to do this ethically. And I'm going to illustrate that here by highlighting work on human genetic diversity that's been carried out over the last 25 to 30 years, beginning with the Human Diversity Project and the Genographic Project, which were both interested in understanding what was the breadth of human genetic diversity. And these projects really targeted Indigenous people groups to understand where those, as described by them, pockets of unique genotypes potentially within the human population that have not been included within larger genetic diversity projects in humans. It was bad enough that the Human Genome Diversity Project was labeled a vampire project by a number of Indigenous groups globally. Those being 25 to 30 years ago, you may suggest that we've learned our lesson, but I'm going to argue now that we have not. And this is best illustrated by a number of current and ongoing projects to again assay human genetic diversity here with the United States that are continuing to shirk the responsibility and communication with Indigenous peoples within the larger context of studies. So if as a community, we've been unable to do this here, I think it's going to be a challenge for us to be able to continue to do that into the future. And that occurs not only for the biological resource, but it also occurs within data. As data moves away from tribal participants, the ownership of that data begins to shift heavily and heavily away from them and towards government and academic institutions. This has been best illustrated by a number of new NIH policies rolling about around data availability and open access, but have existed for quite some time. And that becomes particularly problematic because tribal participants are not single individuals, but within Canada and the United States, they represent different types of legal entities. Specifically within the United States, there are governance and sovereignty components that are inherent to federally recognized tribal groups and some state recognized groups. And so we're not dealing with individuals, we're really dealing with a nation state here when we're talking about tribal or Indigenous participants within larger projects. So to apply the recommendations that were given on the previous slide, we can continue attempting, but mostly failing to do that within current academic frameworks, which are fundamentally tribal trust relationships. We can try and shift these to tribally driven research, where tribes carry out their own research projects funded by their own infrastructure and resources. There are a number of failings that I think are going to be problematic there, including staffing and the ability to ensure continuity. So what I'm going to propose here is operating under tribal partnerships with research entities that exist under tribal governance on tribal lands, but are independent of tribal governments such that turnover within tribal governments don't affect their operations to ensure continuity and the ability to compete for grants and other resources to recruit and retain researchers who are Indigenous to carry out the studies, understanding the context and the frameworks, as mentioned before, that can be complex within biodiversity research. And a really nice example of that is a Native Biodiversity Consortium that was founded about 4 years ago on the Cheyenne River Sioux Tribe as a 501c3 organization. It has the capacity to carry out a number of different types of research, including microbial ecology research. Those microbial projects have been centered on things of interest to communities. So there's direct continuity between individual interest within the community and the benefit that's going to be derived back to the community. Highlighted here by the studies of autoimmunity in the Lakota, the SAIL study, which is looking at origins of increased incidence of rheumatoid arthritis and autoimmune disorder within residents of Cheyenne River. And our focus within this is specifically understanding whether or not there are microbial components that are contributing to this. The second thing I want to highlight is another tribally driven research study trying to understand how land use changes the ecology of the environment based on those practices. And this is being done by sampling cores of soil used for different purposes, either prairie used for growing and collecting hay, or pasture used for grazing. And as you can see here in these two pictures, which were taken 30 feet apart from each other, there are very different environments based on the grass types that are growing, as well as the soil composition, the microbes that exist within the soil. These studies operate within ecological frameworks that are best understood by the indigenous people that are involved and best carried out so that the data remains within indigenous hands so that it benefits indigenous people directly. So there's clear tribal oversight in the approaches that are taken, how the research is being carried out, the interpretations, and the benefits that comes from performing that research as well. So with that, I'd like to acknowledge a number of different groups that have helped contribute to building out these and other frameworks around moving the power dynamics that exist within research from academic entities towards tribal hands, including the Native Biodata Consortium, the Indigidata Workshop, and the SING Consortium here in the United States, as well as a growing body of indigenous microbiome scientists. Thank you all.